Hello, I'm Chaoyu from UT Health San Antonio. I will introduce our recent publication in JEM titled TGF-beta promotes stem-like T cells via enforcing their lymphoid tissue retention. We were interested in the function of TGF-beta signaling in CD8 T cells. Using a conditional mouse line lacking TGF-beta receptor 2 in T cells and adaptive co-transfer system, we examined the response of wild-type and TGF-beta receptor deficient CD8 positive T cells to chronic LCMV infection. First, we found that without TGF-beta receptor, Tcf-1 positive stem-like CD8 positive T cells was significantly reduced, especially at later stages after infection. Interestingly, we also discovered that wild-type Tcf-1 positive T cells mainly express integrin alpha-4 beta-1 with minimal alpha-4 beta-7 expression. In contrast, wild-type Tcf-1 negative T cells carry both alpha-4 beta-1 and alpha-4 beta-7. In the absence of TGF-beta receptor, the expression of alpha-4 beta-7 was significantly enhanced, while alpha-4 beta-1 was slightly reduced. Importantly, the change in alpha-4 integrin can be detected around day 7 to day 10, before dramatic changes in Tcf-1 positive cells occur. Since integrins are involved in cell migration, we found that TGF-beta receptor 2 knockout cells showed enhanced circulation in the blood. When focused on T cells inside the spleen, we discovered that in the absence of TGF-beta signaling, there was a significant reduction in tissue resident markers CD69 in both Tcf-1 positive and negative cells. For wild-type cells, Tcf-1 positive subset is largely confined to lymphoid follicles, as demonstrated by the lack of intravenous anti-CD8 labeling. In contrast, TGF-beta receptor 2 knockout Tcf-1 positive cells show dramatically enhanced intravenous CD8 labeling, suggesting their defects in lymphoid tissue retention. To test whether altered alpha-4 integrin expression is the cause or consequence of reduced Tcf-1 positive cells, we blocked alpha-4 integrins after the initial phase of the infection. We observed partial but significant rescue of Tcf-1 positive subsets in TGF-beta receptor 2 knockout cells. Importantly, alpha-4 integrin blocking also boosts wild-type Tcf-1 positive cells. To determine the immediate effects of alpha-4 blocking, we shortened the treatment. Strikingly, after 2 anti-alpha-4 blocking antibody injections, we detected dramatically enhanced trapping of Tcf-1 positive subset inside lymphoid follicles. Together, our results support a model that TGF-beta dependent lymphoid residency is required for the optimal maintenance of Tcf-1 positive CD8 positive T cells. The expression of alpha-4 integrins is an important component of this residency program. Leaving lymphoid niche will lead to effector differentiation.